Okay, we're starting a new chapter 3, molecules of life. Still doing some chemistry, but now rather than the real basic chemistry, we're going to be talking about the medium and larger molecules to make up cells, make up living things. Okay, was that good? So starting off with lactose, we'll be talking about sugars, amino acids, nucleotides, and then the larger macromolecules of life, proteins, carbohydrates, lipids, and nucleic acid. So lactose is a sugar, major sugar found in milk, and lactose intolerance, inability to produce, to properly digest lactose. And there's an enzyme, and we haven't got the enzyme yet. There'll be in a later chapter talking about the working cell. And enzymes speed up chemical reactions. We're going to have a whole lab on that. In fact, the first on-campus lab next month is going to be on enzymes. And you have to break down lactose in order to be absorbed into the small intestine. Because it's a disaccharide, it's two sugar molecules together, and you can't absorb a disaccharide. You have to absorb only what are called monosaccharides. So you need an enzyme to break it down, and people. And here's the interesting thing about lactose intolerance. And later on, we'll get into genetics. We learn that enzymes are proteins, and most enzymes are proteins. And the recipe for a protein is found in a gene. And so you read genes, you express genes to make proteins. So here's the interesting thing about lactose intolerance. Are they missing the gene for the lactase, the enzyme to break down lactose? No, they're not. Everybody's got that gene, and it works on just about everybody. The thing is, because we are mammals, and as mammals, that's our first food, is mother's milk. If you weren't able to digest milk, the lactose in milk, little babies would starve to death. I'm talking about thousands of years ago or so, when you couldn't get formula, you'd have to rely on mother's milk. The thing is, though, prior to I don't know, hundreds of years ago, this gene would be turned on, and infants would have lactase, digested lactose. And once they were weaned, started eating solid food, and were no longer dependent upon mother's milk, that gene is turned off. And then, even though they have the gene stop turned on, so they don't have any lactase, so they can't digest lactose. But somehow in the Middle East, sometime, I'm not sure when, somehow in some people, this gene was not turned off. It stayed on, so it could digest milk as an adult, and they became a little healthier because of that, because they had a food now they couldn't have digested before. And this gene and this mutation that kept this gene on spread throughout the entire human species, and now almost everybody can digest lactose, except for some people where it still hasn't turned on. Because we are really a strange mammal, really strange, because no other mammal drinks milk once they're weaned from mother's milk. They never touch stuff ever again. And what's even stranger is no mammal drinks the milk of another mammal. It's just, human milk. But we drink cow's milk and goat's milk, which is really unusual. Like I said, no other mammal does that. Okay. So Here's why macromolecules matter. Okay, so you can avoid, now you can buy all kinds of stuff. If you're lactose intolerant, you can buy all kinds of stuff. Now, dairy foods and cheese and everything that have no lactose in them. They take the lactose out, so that's not a big deal. Okay, organic compounds. The cell is mostly water. The rest of the cell contains mainly of carbon-based molecules. Carbon forms large, complex, and diverse molecules, and organic molecules are carbon-based. Here's the thing. We're often called carbon-based life because our big biological molecules have carbon skeletons. They all have carbon skeletons, and so carbon is incredibly versatile. It can form all kinds of different shaped molecules and can combine with just about every other atom on the periodic table. But organic compounds are carbon-based molecules. There's one other thing. An organic molecule is carbon-based, but it also has to have at least one carbon-hydrogen bond. All right? It can be carbon-based, like carbon dioxide, CO2. But CO2 is not an organic compound because it has to have a carbon-hydrogen bond. Sugar, that would be a carbon, that would be an organic compound. Okay. Like I said, it's a versatile molecule, can share electrons with other atoms in 4 covalent bonds. It forms 4 bonds, right? Why? Because it has 4 valence electrons. And it wants how many? 8. So 4 from 8 is 4. So it makes 4 bonds. It always makes 4 bonds. And it can combine with other carbon atoms to make all kinds of different size molecules and different branching patterns, circular molecules, all kinds of different carbon skeletons are possible. There's some carbon skeletons right here. Carbon skeletons vary in length. They can have double bonds or single bonds. They can be branched or unbranched, and they can be circular. That's all a possibility. So the other atoms that are really important in making these big biological important molecules are hydrogen, oxygen, and nitrogen. As a matter of fact, in the Molecular Models lab, we will be making, it's going to be a virtual lab, so we believe we'll be making these models of biological molecules with only containing those 4 atoms: carbon, hydrogen, oxygen, and nitrogen. 90-something% of your body is composed of just those 4 atoms. And then there's a bunch of other smaller ones, I think there's like 26 different atoms found in the human body, some of them in very, very tiny amounts, like the trace elements. But the big 4, carbon, hydrogen, oxygen, and nitrogen. Okay. Methane, CH4. It's an organic compound. Why? Because it has carbon and at least one CH bond, a covalent bond between carbon and hydrogen. Abundant natural gas used by prokaryotes that live in swamps, in digestive tracts of grazing animals like cows. It's like a form of natural gas. At standard temperature and pressure, it's a gas. Larger organic compounds are the main molecules in the gasoline we burn in cars and in other machines and are potent fuels in your body. As a matter of fact, we're going to later on in the later chapter, we'll talk about how fuel molecules are burned in cars and how they are burned in human beings, and it's pretty similar. It's just a lot more slowly, more controlled in living things. It's called oxidation, stripping off electrons. In a car or in a fireplace, you strip off electrons all at once and you form a flame. In living things, the electrons are stripped off more slowly in more controlled ways. Okay, the energy-rich parts of fat molecules have a structure very similar to gasoline. They're called hydrocarbons. They're almost all just carbon and hydrogen, no nitrogen, and very little oxygen, and very big non-polar molecules, very similar to gasoline. So there's gasoline on the left, just carbon and hydrogen, long chains of carbon and hydrogen. Dietary fat called triglyceride on the right, lots of long chains of carbon and hydrogen. A few little oxygen molecules, the red ones there. They're very few. They look pretty similar. Although you drink gasoline, you're going to kill yourself. So we can't use gasoline for fuel in living things, but we use dietary fat, similar, but we're going to find out that even very, very small changes in large molecules may have big biologically different effects. Right? The shape of molecules is really important. Okay. The unique properties of an organic compound depend upon that carbon skeleton and the atoms attached to the skeleton. And the other atoms, the hydrogen, the oxygen, the nitrogen, are formed these different structures called functional groups. And we're going to go over the functional groups. And these functional groups are attached to the carbon skeleton. And the way they're attached, and which functional groups and the way they're attached, determines the biological properties of these molecules. And there they are, functional groups. Groups of atoms directly involved in chemical reactions are functional groups. Each functional group plays a particular role, and a lot of biological molecules have two or more functional groups. So on a molecular scale, many of life's molecules are gigantic. They're really big molecules. We're going to name macromolecules. And the three categories of macromolecules, I would put in a fourth category. They're going to give you life's larger molecules. Again, I'm not sure whether they leave out lipids. I would consider lipids macromolecule. Also carbohydrates, proteins, nucleic acids. You may have heard about these before. We'll be talking a little bit about nutrition, because these are the molecules that we eat and that we derive energy from. Okay. Oh, I see. This is why. Macromolecules are polymers. I would say, yeah, but not all macromolecules are polymers. So this is why I think they leave out lipids, because lipids are not polymers. Right? Polymers are long strings of either exactly the same molecule, as in carbohydrates, or a very similar molecule, like in proteins and nucleic acids. So there are smaller molecules called monomers, medium-sized molecules that are put together in long strings to form polymers. Right? And so 3 out of these 4 macromolecules are biological polymers. Okay, and we put them together what's called a dehydration reaction. And what does dehydration do? It takes water away. So we're going to remove a molecule of water to attach one monomer to another monomer to another monomer. So a very short polymer, you have a monomer, and you always have OH groups hanging off of these monomers. And what happens is an OH is taken off of one monomer, and H taken off the other monomer, and that forms H2O water. You see? So that's how you build a polymer chain, by adding monomers in successive dehydration reactions, leaving you with a molecule of water. Okay, organisms also break down macromolecules. So we make macromolecules, we break them down. Digestion breaks down macromolecules. So you take carbohydrate and you digest it into individual sugar molecules, and those you absorb. And this is called hydrolysis. It's actually the opposite of dehydration. Dehydration makes a molecule of water. Dehydration breaks water and adds the OH and H to the to two broken monomers. And it reverses the dehydration reaction, so it breaks things down. Matter of fact, hydrolysis means breaking of water. Hydro is water, lysis is breaking. So there we are breaking a polymer chain. We're adding a molecule of water to break apart this polymer. So here are the four categories of large biological molecules. So I would call them macromolecules. They're not. They only call them the macromolecules the ones that are polymers. But anyway, carbohydrates, lipids, proteins, and nucleic acids. So they're saying carbohydrates, proteins, and nucleic acids are macromolecules. These are four categories of large biological molecules. All right. So three of them are macro. Okay. Carbohydrates include sugars and polymers of sugar. As a matter of fact, we'll talk later about food labels. In food, there's what are called simple carbohydrates and complex carbohydrates. Simple carbohydrates are actually sugar. A lot of times, disaccharides, that is two sugar molecules attached to each other, or actually just monosaccharides. That one single monomer is called a simple carbohydrate. Complex carbohydrates are long chains of many monomers, sugars together to form what's called a polysaccharide or carbohydrate. So remember, monosaccharide is one sugar molecule. Disaccharide is two sugar molecules. A polysaccharide is a long chain of sugar molecules. And a polysaccharide is the same as a carbohydrate. Okay, so small sugar molecules found in soda and large starch molecules found in spaghetti and bread. So the small sugar molecules, simple carbohydrates. The starch in spaghetti, bread, potatoes is a complex carbohydrate. So in animals, carbohydrates are a primary source of dietary energy. That is short, and mostly it's like a short term, from meal to meal. You're living both off of fat and carbohydrates, but usually think of carbohydrates as sugar as short-term energy and dietary energy, and also they serve as the raw material for manufacturing other types of molecules. You'll find out that these sugars can be converted into lipids, for example, and it can be stored as fat. So in plants, carbohydrates serve as a building material for much of the plant body. Now, there's different carbohydrate, we're going to get into that, called cellulose, that makes up the cell wall of plants. And that's really like this, almost like the skeleton in the plant that holds it together. We have an internal skeleton of bone. Their skeleton is really carbohydrate. The cell walls around their cells give them strength. So monosaccharides are the monomers of carbohydrate and cannot be broken down into smaller sugars. Common examples are glucose in spore strength and fructose found in fruit. Both these simple sugars are also found in honey. And in fact, glucose is probably the most important as far as our nutrition goes, and nutrition for a lot of other animals, because glucose is our blood sugar. When we talk about blood sugar, we talk about glucose. And actually, any other monosaccharide you take into your body is actually converted to glucose. So here are two what are called isomers. They have this, okay, we talked a little bit about this in the last chapter, and we'll talk about it in the lab, in the molecular models lab, about molecular formulas and structural formulas. Molecular formulas are what atoms are found in a molecule and how many of them there are. Glucose, glucose's molecular formula is C6H12O6. 6 carbons, 12 hydrogens, 6 oxygens. Fructose, molecular formula C6H12O6. Exactly the same. But you see how they're arranged different. There's a different arrangement of the atoms. So that's the structural formula. And so molecules that have the same molecular formula, but different structural formulas, are called isomers. And same formula, different arrangements, different structural formula. Okay. And they're going to show you linear, these molecules as linear molecules. That's the last time we're going to do that. They actually form ring structures. And for the rest of the chapter and for the rest of this course, they're going to be, if you see a sugar, it's going to have a ring structure. Okay, so the isomers, molecules have the same molecular formula, but different structural formula. Okay. Okay, we're not going to get into structural isomers, geometric isomers, and enantiomers. Don't worry about that. We're not going there. Okay. You do have to just know what an isomer is. Okay. So monosaccharides, particularly glucose, are the main fuels for cellular work. In water, monosaccharides form ring structures. That's why we, like I said, we're going to move to a ring structure. Like this. So there's the linear structure on the left, and there's the ring structure in the middle. As a matter of fact, sometimes you just really abbreviate it and just make this hexagonal ring-like structure where all the angles are carbons. You just take it for granted that an angle is a carbon and that there are OH and H's hanging off of those carbons. Each carbon makes 4 bonds. Right? To abbreviate it. All right. Linear ring structures. Okay. Disaccharide is a double sugar constructed from 2 monosaccharides by a dehydration reaction. In some, like we were just talking about lactose, that's a disaccharide. We look at sucrose, which is regular table sugar, also a disaccharide. And maltose, which is really like grain sugar, is also a disaccharide. So lactose is a disaccharide between glucose and another monosaccharide called galactose, which never actually exists by itself. It's always attached to glucose to form lactose. And when you drink milk or dairy products and you get the lactose in it, you break it apart and you absorb glucose and galactose. And galactose is converted by your liver into glucose. And so notice that it is a dehydration reaction. We remove water from the 2 molecules and we put them together with the oxygen in between them. As I mentioned, disaccharides include lactose in milk, maltose in beer, sucrose in table sugar. So sucrose is the main carbohydrate in plant sap, and also the main sugar in corn is glucose. And here's the thing: we used to companies used to make sugary drinks like sodas and everything else using sucrose that comes from sugarcane, but sugarcane is not grown in the U.S. Although there might be some in Hawaii, but it's a tropical plant, so it's not grown in the U.S. So we have to import all of it. But we have lots of corn. I mean, we got lots of corn. We grow enormous amounts of corn, and corn is sugary. It's full of carbohydrates, so obviously it's got sugar. So carbohydrates are long polymers of sugar, glucose. So the corn sugar is glucose. Here's a problem though. Even though glucose is a sugar, we usually think, when you think of sugar, oh, sugar is sweet. Of course it is. But actually, glucose is not all that sweet as sugars go. And it has to do with actually our taste receptors. Some sugars really activate taste receptors and get this intense sweetness. Other ones don't really act them that much. And it's not, so glucose is not that sweet. So we want to make it much sweeter. So what they do is they take perfectly good corn sugar, they bring it into a lab, and they make high fructose corn syrup, HFCS. And it's a commercial process. It's made in a lab. I try to avoid this stuff because it's just me. I'd rather, eat something or drink something that's made by some other organism and not made by some chemist in the lab. Anyway, so it's much cheaper and it's produced here. So now almost all sugary drinks are full of high fructose corn syrup. It's often the first ingredients listed in soft drink. Except for things like retro Coke, where they put sucrose back into it. Okay, and there it is, corn. Processed to extract starch. The starch is broken down to glucose. Glucose is converted to fructose, added to soda as a high fructose corn syrup. And here's the thing, we have a bit of overnutrition for mostly in the United States and putting on weight and getting extra calories. And the average American consumes about 45 kilograms of sugar per year, mostly as actually high fructose corn syrup. Is a major cause of tooth decay. Overconsumption increases the risk of developing type 2 diabetes. And we have an epidemic of type 2 diabetes in this country, and heart disease. You'd be amazed at the amount of extra sugar added to stuff. If you just read food labels, you see, wow, it's put in yogurt and breads and of course the sugary drinks. It's all over the place. You find sugar, lots of added sugar. So polysaccharides, as I mentioned, complex carbohydrates made of long chains of sugars, polymers of monosaccharides. And we're going to look at various types of carbohydrates, starch being one of them. Long strips of glucose monomers used by plant cells to store energy, and potatoes and grains are major sources of starch in our diet. As a matter of fact, most nutritionists think that the most of the calories they get during the day should be from carbohydrates. So starch, it's a plant product. There's starch granules in potato tuber cells. See, it's a long chain of glucose monomers. Now, glycogen is another carbohydrate, sometimes called animal starch, because it's used by animal cells to store energy. We eat the potato, we break it into glucose, we absorb it, we send it to our liver. The liver makes glycogen out of it. And then in between meals, the glycogen is broken down into glucose, goes into the blood, and it maintains blood sugar between meals. So it's broken down to release glucose when you need energy to maintain blood sugar, because that's a homeostatic variable in your body. Blood sugar, and always has to be at the right level. Okay. There's glycogen granules in muscle tissue. So it's either broken down by the liver and stored in the liver. It's also stored in muscle. Then cellulose, most abundant organic compound on Earth, forms cable-like fibers, and it is the cell wall of plants. And it is a carbohydrate that animals cannot break down. We can't break down the stuff. It's indigestible carbohydrate. It's fiber. It's called insoluble fiber. So when you eat, and some plants are almost 100% cellulose, like a celery stalk. You eat a celery stalk, you get a little bit of nutrient, but mostly it's like 98% cellulose. And you can chew it as much as you want. This is called mechanical digestion. But you're not going to break it down enough for you to absorb any nutrients from it. So it goes in looking one way, it comes out the other end looking really different, but it's virtually the same thing. It's cellulose. We cannot digest it. Although it's really healthy to eat, despite the fact that it's not digestible. And we'll probably talk about that later on. Why they always tell you, oh, you should eat your fiber, get lots of fiber. Even though it's non-nutritive, you don't get any calories from it. It can't be broken down by any enzymes produced by animals. And that includes termites, cows, horses that eat grass that's almost 100% cellulose. So how can they live off of that? Well, they have bacteria in their guts, actually in the gut of termites, it's a protista, that has the enzyme to break down cellulose, it's called cellulase. They can break it down. So they are symbiotic organisms inside cows and termites and horses and goats that break down the cellulose for those animals. Right? And there's the cellulose microfibrils in plant cell wall, and cellulose molecules. See, they're long chains of glucose, almost identical to starch, but the bond between the sugar molecules is different in cellulose than in starch. It's actually called alpha bonds in cellulose and beta bonds in starch. Beta bonds, we got the enzymes to break. Alpha bonds, we don't. Okay, on that note, we'll leave carbohydrate, and so I'm going to call it here for this. I guess this will be 106, chapter 3. 106 life molecules 1, I guess. Well, anyway, I'm going to call it here, and we'll pick it up in the next video when we start talking about lipids. Okay, so I'm going to stop recording now.